Welcome to this presentation. My name is Clara Inserte and I am a medical informatics PhD student at the University of Münster. For this poster, I compared multiple integration strategies in fixed RNA profiling for a comprehensive tumor analysis. So first, let's start with a brief introduction. In cancer research, exploring the similarities and differences between distinct tumor entities is essential to advance our understanding of the disease. For this goal, single-cell analysis has been increasingly used. A recent advancement in this field of single-cell analysis is fixed RNA profiling, which enhances efficiency and scalability of sample processing by enabling extended storage and batch processing. The bioinformatics analysis of this data mandatorily requires the integration of samples to eliminate disrupting batch effects. However, the standard approach that's followed in single-cell analysis, which is considering each sample as an independent experiment, may lead to overcorrection when applied to fixed RNA data. An ideal integration approach successfully removes all batch effects while preserving biological differences. And while previous studies have evaluated different integration methods for single-cell transcriptomics data using healthy tissue as a benchmark, the applicability of these findings to tumor tissue and fixed RNA data remains uncertain. For this comparison, we performed fixed RNA profiling using the 10x Chromium technology. We sequenced 44 chorioid plexus tumor samples and 33 posterior fossa ependymoma samples. Each of the datasets contains 3 subtypes. Figure 1 here shows the workflow of 10x Chromium technology for fixed RNA profiling. Samples are processed in batches, resulting in less variability between samples from the same batch. Therefore, an integration only taking into account batches as independent experiments might be better for the analysis of this data. Table 1 shows an example of the metadata for each sample. We have the information on the batch each sample was processed in and which subtype it belongs to. With this information, we defined a new annotation called combined annotation, as it combines the information on batch and subtype labels. We compared 3 different algorithms: harmony, canonical correlation analysis, also called CCA, and reciprocal principal component analysis, also called RPCA. And then 3 different strategies, which are considering each sample as independent experiment, each batch as independent experiment, or the combined annotation of subtype and batch as an independent experiment. We also compared the results to the merged object, which is the one without any integration. All the pre-processing of the samples was done identically for all cases, following the SURAT version 4 guidelines. In the following results, I'll show some examples of the combination of strategies that we compared. These first 2 UMAP plots show the distribution of the cells in the UMAP space after merging all samples into a single object without integrating to correct batch effects. We observe that the subtypes are quite separated, but also there's a lot of batch effects, as some clusters only contain cells from a single batch. Integrating the data using the CCA algorithm and considering samples or the combined annotation as independent experiments leads to what seems an overcorrection of the batch effects, as after this we lose all variation between the different tumor subtypes. Considering batches as independent experiments and using harmony or RPCA methods to integrate yield the best results in terms of batch effect correction while preserving the biological differences between tumor subtypes. As we can see here, batches are mostly overlapping for all clusters, while we keep the variation between the tumor subtypes, meaning there are some clusters that only have cells from one subtype. And on this slide, we see the example of integrating using harmony and considering batches as independent experiments. The results are similar to the ones before, even maybe a bit better in terms of batch effect correction. As a general conclusion, we can say that these comparisons provide valuable insights into the selection of integration strategies, paving the way for enhanced accuracy and reliability of fixed RNA data analysis across diverse tumor types. And here, to finish this presentation of my poster, are the references that I used to carry this comparison out. First of all, the previous benchmark for these integration methods, and the next three references are the papers where these methods were published.